Good afternoon, everybody. This is Ron Firestein from the Hudson Institute of Medical Research in Melbourne, Australia. And I'll be talking today about mapping the therapeutic vulnerabilities in molecularly defined pediatric brain cancers. So, as we all know, over the past 10 years or so, there have been large scale efforts that have provided us a deep understanding of the molecular landscape of the pediatric brain cancer genome. And this includes groups such as CBTN, St. Jude's, DKFZ, and others that have provided a treasure trove of information regarding the molecular alterations, such as mutations, DNA methylation, pathway perturbations, and has enabled us to classify pediatric brain tumors at a very comprehensive molecular level. However, it's important to note that we still don't know the functional implications of the vast majority of these alterations. And in order to ascribe such function, we really need to think about an integrative approach that not only combines these types of tumor molecular sequencing exercises, but also utilizes clinically relevant models that recapitulate the molecular features of the tumor, together with functional approaches such as chemical perturbants, such as small molecule libraries, and functional genomics, RNAi, and nowadays CRISPR, to really identify which of these alterations are actually drivers. So this type of integrated approach has been successfully employed for adult cancers, and many of us are very familiar with the Sanger Institute's cell line project and the Broad Institute dependency map, which have characterized over 1,000 cell lines across different adult malignancies. However, in pediatric brain cancers, this has really been woefully underrepresented in these kinds of efforts. And you can see both the number of models is much lower, as well as the characterization using functional genomic or drug screening approaches has really significantly lagged the adult counterparts. So with this in mind, we started a new program about 4 years ago that we call the Childhood Cancer Model Atlas. And this program has three main goals, as you can see here. The first, to develop a well-annotated living biobank of well-characterized pediatric cancer lines, and to use this type of repository to identify new targets, pathways, and biomarkers, and generate hypotheses that will enable us to define the biological pathways that comprise these kinds of cancers. And lastly, to accelerate the implementations of these new therapeutic opportunities into the clinic. So we quickly realized that we couldn't really do this on our own in Australia. This type of effort would really require an international global partnership with many organizations. And I think you can see here that the Children's Brain Tumor Network in particular has been a fantastic resource for us, and a great partnership, both in terms of providing these models, but also at the intellectual level as well. And together with CBTN and many other partners, we've now collected and processed over 450 pediatric models. And it's important to note that the vast majority of these are actually from CNS tumors. So you can see here in this schematic, what are we actually doing with these models? And that includes both analyzing the different omics features, including whole genome sequencing, transcriptomics, epigenomics, as well as at the protein level, both total and phosphoproteomic analysis. And then that's merged with functional approaches such as CRISPR-Cas9 screens, which we've performed about 120 so far to date, as well as high throughput drug screens, to be able to integrate both the molecular features with the different dependencies. So we've made this information publicly available on our website, and you can see the link is shown here at the lower left corner. The numbers are continuously updated. We currently have over 300 active pediatric lines. And importantly, we've provided multi-omic analysis on more than half of these lines, including the different modalities that are shown here. Uh, I'd like to point out that more than two-thirds of these lines, so about 240 lines, are actually from pediatric CNS tumors. And why do we really need this many cell lines is something that some of you might be asking. And I think that's really exemplified by this screenshot from the latest WHO classification of pediatric gliomas. And we're well aware that over the last 5 years or so, the diagnostics of pediatric cancers, and particularly brain cancers, has become more and more molecularly defined. And that's exemplified here with the diagnosis of high-grade gliomas now comprising molecular information such as H3 mutations and alterations, as well as other altered genetic pathways. So with this type of cell line atlas, we can really provide us an excellent opportunity to model these different molecular classes of pediatric gliomas. And that's really shown here in this oncoplot across our cohort of lines. You can see that indeed we're able to now comprise the major subtypes of childhood brain cancers at a molecular level, and that includes the H3.1, K27M DMGs, likewise A3.3s, the G34 diffuse hemispheric gliomas, the wild types, and we also have a cohort of adult gliomas as well. So with this type of cohort, we can now begin to ask really interesting questions like what are the um, differences, the functional genomic differences between childhood and adult gliomas, as well as get down into more granular details looking at diffuse midline versus diffuse hemispheric gliomas, and likewise even more granularities around looking at differences between the different histone mutant subtypes. So really these types of questions are informed by our growing cohort of lines that we've been able to subject to CRISPR-Cas9 screens. And using these types of screens, we can now compare genetic dependencies between adult and pediatric gliomas. You can see here in this scatter plot, the adult glioma-specific genetic dependencies are color-coded in purple, while genes that are specifically important in pediatric gliomas are color-coded in green. And I think one of the really exciting aspects of this is exemplified here with genetic targets such as MCL1, PIK3CA, and epigenetic factors that already are druggable, which gives us a kind of an early lead to potentially testing whether inhibitors to these genetic pathways might be able to progress into the clinic. And just to illustrate one example of this, one of the most striking dependencies that we really found in our pediatric glioma cohort was in the PI3 kinase pathway. And you can see here the PI3 kinase gene PIK3CA was really important in the vast majority of the pediatric gliomas color-coded here in green across this waterfall plot. You can see that greater than 80% of our pediatric gliomas were exquisitely sensitive to PIK3CA knockout compared to adult gliomas, which were much less sensitive. We also know that in adult cancers, PIK3CA mutation is a very important marker in predicting response to these PI3 kinase inhibitors, and that's well described and also shown here in this recent New England Journal of Medicine paper showing the activity of alpelisib, an FDA-approved PIK3CA specific inhibitor. And the data clearly shows that the alpelisib combination worked very well in patients that had PIK3CA mutant breast cancer. But interestingly, what we find is that this was not the case in pediatric glioma. So, in general, both adult and pediatric brain cancer harbor PIK3CA mutations at a relatively low frequency of about 15%. However, you can see that unlike adult cancers, where PIK3CA mutation clearly predicts response to um, the PIK3CA knockout, you can see that in our pediatric gliomas, we found that both PIK3CA mutant and wild types were equivalently dependent on this particular gene. And we wanted to then test whether this might be translatable using inhibitors. And indeed, we tested alpelisib, an FDA-approved and selective PI3 kinase inhibitor that's already being used for advanced breast cancer, and also importantly improved in children with the um, PI3 kinase related overgrowth syndrome. So indeed, we were able to validate our genetic dependency findings using this drug inhibitor, which clearly showed that the uh, PI3 kinase wild type and mutant pediatric gliomas were equivalently sensitive to alpelisib compared to adults, where only the PIK3CA mutant adult glioma was sensitive compared to the wild type cell lines. We then turned our attention to look at our cohort to see whether the H3 altered subtypes of gliomas correlated with unique genetic dependencies. And I was particularly excited because that has not been reported yet in the literature. And the way we approach this is by defining a residual score, which takes into account both the strength of the genetic dependency and the frequency in the cohort. And using this approach, you can see here that what really bubbled to the top were genes that were important in epigenetics or transcription in the H3K27M altered DMG cohort. Likewise, in the H3G34 diffuse high-grade gliomas, interestingly, we found enrichment in genetic dependencies, and particularly in protein kinase-related signaling pathways. So the biology of this remains to be worked out, but I think the important take-home message is that indeed we are able to find unique genetic dependencies across these different H3 altered subtypes. So lastly, in the current WHO, H3K27 altered DMGs is considered a single entity, but as we well know, the H3K27 mutations can exist in either H3.1 or the histone H3.3 protein. And it's important to note that these tumors do show differences in clinical features such as location, age, survival profile, and also in co-occurring genetic alterations. So we then asked whether there were indeed functional differences in between the H3.1 and H3.3 cohort. And indeed, we found that there was. And just to show one particular example that we think is quite interesting is that you can see that HDAC2 was particularly important in the H3.3K27 altered line. And in this density plot, you can see in light green are the 3.3 mutants, which the vast majority of which were exquisitely dependent on HDAC2. You can see that this is specific for HDAC2, as HDAC1 itself was not important across either cohort of H3 mutant gliomas, and HDAC3, which I don't show here, but is essential actually in across all the gliomas. So clearly there was this very specific and selective effect for HDAC2 dependency. And importantly, we were able to validate that using single guide RNA knockouts. And what you're looking at here are studies where we knock out HDAC2 and then reintroduce either the wild type or a mutant version, catalytically dead version of HDAC2. And importantly, we found that only restoration of the HDAC2 wild type was able to restore growth after knockout, showing us that indeed the enzymatic activity of HDAC2 is particularly important for the growth of these lines. So lastly, we wanted to go beyond the genetic dependencies, and with the molecular features that we've compiled across all these lines, we wanted to ask whether we could identify particular biomarkers that predict these genetic dependencies, and two, whether these biomarkers were the same or different between adult and pediatric brain cancers. So in this scatter plot, you can see genes whose dependencies can be predicted by a set of molecular features using a machine-based learning algorithm called random forest. And importantly, the ones marked in orange here are genes that have a set of features in both pediatric and adult cancers. But the question we really wanted to answer is, were those features the same or were they different? And interestingly, the answer is that in about half of the cases, pediatric cancers did have unique biomarkers that predicted response compared to the adult cancers. And just to illustrate this in a little bit more detail, to exemplify this, you can see one example, MDM2 dependency is really predicted by the P53 wild type status of the lines, and that's really the same across both adult and pediatric cancers. However, in other cases, we found, such as in MCL1 genetic dependencies, it's well described that BCL201, another compensatory anti-apoptotic gene, is very important. Its expression is important in predicting MCL1 dependency. And this was certainly the case in adult cancers, but indeed in pediatric cancers, BCL201 expression did not play an important role, suggesting that indeed, in certain cases, there are unique biomarkers in pediatric gliomas that should be considered as potential predictors of response, and they would be different from the adult cancers. So just to summarize, what I've shown here is the development and characterization of a childhood cancer atlas, really focused on pediatric brain cancers. And this provides a collection of diverse models representing the deadliest childhood cancers, enabling a streamlined process towards identification of therapeutic targets and potential biomarkers for their response. And we also show the development of an online database to provide both the existence and characterization of these models. So using this approach, two important take-home messages were that indeed we found that pediatric high-grade gliomas do harbor distinct functional drivers of growth. And importantly, that these gliomas have also distinct biomarkers of response, even across shared adult and pediatric genetic dependencies that may spur the development of pediatric-specific biomarkers and not just therapeutic targets. So for the future, we continue to develop new models, particularly to rare types of pediatric brain cancers, such as ependymomas, ETMRs, choroid plexus carcinomas. And if you have such models and want to collaborate with us, please do get in touch. We are expanding our repertoire of omics to metabolomics as well, and continue to expand our drug and CRISPR screen approaches to really be able to put together a large data set to predict which patients will respond to particular types of therapies in the future. I'd obviously like to thank our large team in Melbourne and our fantastic collaborators, the CBTN group, particularly Mateus and Adam, who have been um, really cheering us and helping us along the way with cell lines and intellectual support, and all our collaborators who have contributed to this project. Funding partners are the Children's Cancer Foundation, Robert Connor Dawes, and the Australian government. Thank you very much.